So let's talk about muscle contraction. Before we dig into the steps, I'd like to remind you about an activity we did first week of HAPS class where we were modeling a impulse conducting through a neuron's cell membrane. And so we stood outside or in our classroom and we had NAs on one of our hands and Ks on the other hand. So that was sodium on one hand and potassium on the other hand. And then your crazy teacher gave a starting initial impulse. So I started the cycle that began this process. And all of a sudden your sodium hands twisted and moved into the cell, and that knocked the potassium hand out of the cell. And then you were twisted at the waist, so you had turned yourself around. And as soon as you did that, the person behind you, simulating the next section of the membrane, did the exact same thing. Once they flipped around, however, you went back to resting position. The potassium went back into the cell and the sodium went back outside of the cell. That was the job of the sodium-potassium pump. So again, initially, you had sodium rushing in at the onset of the impulse. Potassium was forced out, and then after that had triggered a response from your next door neighbor tissue, then the sodium went back out and the potassium went back in. You're going to see something very similar here in the conduction of this muscle. So let's start. A says that you've got a motor cortex is going to, or motor cortex neurons are going to send a move impulse, releasing acetylcholine into the synapse. The synapse is probably going to be right along the sides of the fascicle. So it's not just on the side of the muscle body, it actually is going into the muscle bundle itself and innervating fascicles. And then take a look up here at letter B. It says sodium channels open and sodium rushes in, creating a graded potential in a muscle cell. So that sounds exactly like what we were doing that day outside trying to model what a neuron does. But this is a muscle cell. So that means that we have this very specialized cell that can also conduct this electrical impulse. Then we go down here to letter C. Letter C says that these T tubules allow the impulse all the way through down to the muscle cells. So we're going from fascicle, getting that sodium channel opening and releasing that impulse even farther, and then the T tubules that are a part of our sarcoplasmic reticulum are getting that impulse all the way throughout the cell. Then I mentioned that sarcoplasmic reticulum, that is going to get innervated when that impulse comes through those T tubules and triggers that sarcoplasmic reticulum, which is a big brother or big sister of a smooth or rough ER. It's going to release a whole bunch of calcium, and that's what's going to allow the next step or the contracting phase to occur. So in this model here, we have purple myosin and black thin actin fibers, and the myosin and the actin are going to pull really hard on each other, moving the actin inwards along the midline and drawing those Z lines on the left and the right towards each other, causing that sarcomere to get shorter. But let's go into the details here. So as you recall, step D said that the sarcoplasmic reticulum is going to release calcium. That calcium actually is going to bind to the troponin, which moves the tropomyosin off actin's binding site. So think about actin kind of being covered in this string of proteins, and along that string of proteins, there's a few of these enzymes called troponin. Troponin, when it binds with calcium, will change shape, which actually lifts the tropomyosin off of actin's binding sites. So now the process can actually take place. ATP is hydrolyzed by myosin, which means it's broken into ADP and then a little P, so phosphate. And that's actually going to cause that myosin to stretch. That's getting it all the way open and ready for its big next move. Myosin attaches to the actin, releasing ADP and P, and then pulls really hard on the actin. So that's called the power stroke. The ATP is hydrolyzed in step E2, stretches that myosin, who's now available to snap and bind to the actin, and then as it binds to the actin, it pulls inward hard and drags the actin towards myosin's center. Lastly, that myosin is going to stay in that pulled end position until another ATP arrives and actually allows the myosin to release the actin and go into that ready position or reset position. This will happen repetitively until there is no more ATP available, or until the calcium that was binding to the troponin or the tropomyosin is actually pumped out of that area of the cell and there is no more calcium, so then the troponin will not be there to change shape. It'll go back to its original site and tropomyosin will drop back onto the actin, blocking the spot where the myosin could bond.